Most recent common ancestor. In biology and genealogy, the most recent common ancestor, MRCA, last common ancestor, LCA, or concestor of a set of organisms is the most recent individual from which all the organisms of the set are descended. The term is also used in reference to the ancestry of groups of genes, haplotypes, rather than organisms. The MRCA of a set of individuals can sometimes be determined by referring to an established pedigree. However, in general, it is impossible to identify the exact MRCA of a large set of individuals, but an estimate of the time at which the MRCA lived can often be given. Such time to most recent common ancestor, MRCA, estimates can be given based on DNA test results and established mutation rates as practiced in genetic genealogy, or by reference to a non-genetic, mathematical model or computer simulation. In organisms using sexual reproduction, the matrilinear MRCA and patrilinear MRCA are the MRCAs of a given population considering only matrilineal and patrilineal descent, respectively. The MRCA of a population by definition cannot be older than either its matrilinear or its patrilinear MRCA. In the case of Homo sapiens, the matrilinear and patrilinear MRCA are also known as mitochondrial leaf, Mount MRCA, and Y-chromosomal atom, YMRCA, respectively. The age of the human MRCA is unknown. It is almost certainly less than the age of either the YMRCA or the Mount MRCA, estimated at around 200,000 years. The last universal common ancestor, LUCA, is the most recent common ancestor of all current life on Earth, estimated to have lived some 3.5 to 3.8 billion years ago, in the Paleoarchean. MRCA of different species. The project of a complete description of the phylogeny of biological species is dubbed the Tree of Life. This involves time estimates of all known speciation events, for example, the MRCA of all carnivora, i.e. the MRCA of cats and dogs, is estimated to have lived of the order of 42 million years ago, Miacidae. The concept of the last common ancestor from the perspective of human evolution is described for a popular audience in The Ancestor's Tale by Richard Dawkins, 2004. Dawkins lists concestors of the human lineage in order of increasing age, including hominin, human chimpanzee, hominin, human gorilla, hominid, human orangutan, hominoid, human gibbon, and so on in 40 stages in total, down to the last universal ancestor, human bacteria. MRCA of a population identified by a single genetic marker. It is also possible to consider the ancestry of individual genes, or groups of genes, haplotypes, instead of an organism as a whole. Coalescent theory describes a stochastic model of how the ancestry of such genetic markers maps to the history of a population. Unlike organisms, a gene is passed down from a generation of organisms to the next generation either as perfect replicas of itself or as slightly mutated descendant genes. While organisms have ancestry graphs and progeny graphs via sexual reproduction, a gene has a single chain of ancestors and a tree of descendants. An organism produced by sexual cross-fertilization, allogamy, has at least two ancestors, its immediate parents, but a gene always has one ancestor per generation. Patrilineal and matrilineal MRCA. Mitochondrial DNA, MRCA, is nearly immune to sexual mixing, unlike the nuclear DNA whose chromosomes are shuffled and recombined in Mendelian inheritance. Mitochondrial DNA, therefore, can be used to trace matrilineal inheritance and to find the mitochondrial Eve, also known as the African Eve, the most recent common ancestor of all humans via the mitochondrial DNA pathway. Likewise, Y chromosome is present as a single-sex chromosome in the male individual and is passed on to male descendants without recombination. It can be used to trace patrilineal inheritance and to find the Y-chromosomal atom, the most recent common ancestor of all humans via the YDNA pathway. Mitochondrial Eve and Y-chromosomal atom have been established by researchers using genealogical DNA tests. Mitochondrial Eve is estimated to have lived about 200,000 years ago. A paper published in March 2013 determined that, with 95% confidence and that provided there are no systematic errors in the study's data, Y-chromosomal atom lived between 237,000 and 581,000 years ago. The MRCA of humans alive today would, therefore, need to have lived more recently than either. It is more complicated to infer human ancestry via autosomal chromosomes. Although an autosomal chromosome contains genes that are passed down from parents to children via independent assortment from only one of the two parents, genetic recombination, chromosomal crossover, mixes genes from non-sister chromatids from both parents during meiosis, thus changing the genetic composition of the chromosome. Time to MRCA estimates. Different types of MRCAs are estimated to have lived at different times in the past. These time to MRCA (MRCA) estimates are also computed differently depending on the type of MRCA being considered. Patrilineal and matrilineal MRCAs, mitochondrial even Y chromosomal atom, are traced by single gene markers, thus their MRCA are computed based on DNA test results and established mutation rates as practiced in genetic genealogy. Time to genealogical MRCA of all living humans is computed based on non-genetic, mathematical models and computer simulations. Since mitochondrial even Y chromosomal atom are traced by single genes via a single ancestral parent line, the time to these genetic MRCAs will necessarily be greater than that for the genealogical MRCA. This is because single genes will coalesce more slowly than tracing of conventional human genealogy via both parents. The latter considers only individual humans, without taking into account whether any gene from the computed MRCA actually survives in every single person in the current population. MRCA via genetic markers. Mitochondrial DNA can be used to trace the ancestry of a set of populations. In this case, populations are defined by the accumulation of mutations on them, and special trees are created for the mutations and the order in which they occurred in each population. The tree is formed through the testing of a large number of individuals all over the world for the presence or lack of a certain set of mutations. Once this is done, it is possible to determine how many mutations separate one population from another. The number of mutations, together with estimated mutation rate of them in the regions tested, allows scientists to determine the approximate time to MRCA, MRCA, which indicates time passed since the populations last shared the same set of mutations or belonged to the same haplogroup. In the case of Y chromosomal DNA, MRCA is arrived at in a different way. YDNA haplogroups are defined by single nucleotide polymorphism in various regions of the YDNA. The time to MRCA within a haplogroup is defined by the accumulation of mutations in STR sequences of the Y chromosome of that haplogroup only. YDNA network analysis of YSTR haplotypes showing a non-star cluster indicates YSTR variability due to multiple founding individuals. Analysis yielding a star cluster can be regarded as representing a population descended from a single ancestor. In this case, the variability of the YSTR sequence, also called the microsatellite variation, can be regarded as a measure of the time passed since the ancestor founded this particular population. The descendants of Genghis Khan or one of his ancestors represents a famous star cluster that can be dated back to the time of Genghis Khan. MRCA calculations are considered critical evidence when attempting to determine migration dates of various populations as they spread around the world. For example, if a mutation is deemed to have occurred 30,000 years ago, then this mutation should be found amongst all populations that diverged after this date. If archaeological evidence indicates cultural spread and formation of regionally isolated populations, then this must be reflected in the isolation of subsequent genetic mutations in this region. If genetic divergence and regional divergence coincide, it can be concluded that the observed divergence is due to migration as evidenced by the archaeological record. However, if the date of genetic divergence occurs at a different time than the archaeological record, then scientists will have to look at alternate archaeological evidence to explain the genetic divergence. The issue is best illustrated in the debate surrounding the demic diffusion versus cultural diffusion during the European Neolithic. MRCA of all living humans. The age of the MRCA of all living humans is unknown. It is necessarily younger than the age of either the matrilinear or the patrilinear MRCA, both of which have an estimated age of between roughly 100,000 and 200,000 years ago. A mathematical, but non-genealogical study by mathematicians Joseph T. Chang, Douglas Rode and Steve Olson calculated that the MRCA lived remarkably recently, possibly as recently as 300 BCE. This model took into account that people do not truly mate randomly, but that, particularly in the past, people almost always mated with people who lived nearby, and usually with people who lived in their own town or village. It would have been especially rare to mate with somebody who lived in another country. However, Chang et al. Found that a rare person who mates with a person far away will in time connect the worldwide family tree, and that no population is truly completely isolated. The MRCA of all humans almost certainly lived in East Asia, which would have given them key access to extremely isolated populations in Australia and the Americas. Possible locations for the MRCA include places such as the Chukchi and Kamchatka peninsulas that are close to Alaska, places such as Indonesia and Malaysia that are close to Australia, or a place such as Taiwan or Japan that is more intermediate to Australia and the Americas. European colonization of the Americas and Australia was found by Chang to be too recent to have had a substantial impact on the age of the MRCA. In fact, if the Americas and Australia had never been discovered, the MRCA would only be about 2.3% further back in the past than it is. Note that the age of the MRCA of a population does not correspond to a population bottleneck, let alone a first couple. It rather reflects the presence of a single individual with high reproductive success in the past, whose genetic contribution has become pervasive throughout the population over time. It is also incorrect to assume that the MRCA passed all, or indeed any, genetic information to every living person. Through sexual reproduction, an ancestor passes half of his or her genes to each descendant in the next generation. After more than 32 generations, the contribution of a single ancestor would be on the order of 2-32, a number proportional to less than a single base pair within the human genome. Identical ancestors point. The MRCA is the most recent common ancestor shared by all individuals in the population under consideration. This MRCA may well have contemporaries who are also ancestral to some but not all of the extant population. The identical ancestors point is a point in the past more remote than the MRCA, at which time there are no longer organisms which are ancestral to some but not all of the modern population. Due to pedigree collapse, modern individuals may still exhibit clustering, due to vastly different contributions from each of ancestral population.